Roughly 4 billion years ago, this planet was nothing but rock, water, and gas. No cells, no genes, no metabolism, nothing that could reasonably be called alive by any definition scientists use today. And yet, at some point during that vast stretch of geological time, something in that lifeless chemical soup crossed a threshold it never crossed back from. Molecules began copying themselves. Chemistry began organizing itself into systems that could capture energy, store information, and reproduce. Life, in other words, simply began. How that actually happened remains one of the last great unsolved mysteries in all of science, right up there with the nature of dark matter and the origin of consciousness. For more than a century, researchers have proposed answer after answer, each one capturing a piece of the puzzle while leaving enormous gaps unexplained. Now a new hypothesis, published in the peer-reviewed journal Research and already generating serious discussion across the scientific community, is attempting something almost nobody else has dared to try. It is not just proposing another isolated theory to add to the pile. It is attempting to unify decades of competing ideas into a single coherent framework, built around structures so small that a few million of them could fit across the width of a human hair. This is the story of the nanozymes hypothesis, why it is turning heads among origin of life researchers, and why it might finally offer a genuinely satisfying answer to a question that has quietly frustrated scientists since Darwin's era. To understand why this new hypothesis matters, it helps to understand just how fragmented the search for life's origin has become. Over the past 100 years, scientists have proposed a long list of separate frameworks, each one built around a different chemical starting point. There is the RNA world hypothesis, which proposes that self-replicating RNA molecules were the first true precursors to life, capable of both storing genetic information and catalyzing chemical reactions long before DNA and proteins took over those separate jobs. There is the metabolism first model, sometimes called the iron-sulfur world, which suggests that simple chemical reaction cycles, not genetic molecules, were the true starting point, with genetics arriving only later. There is the lipid world hypothesis, centered on self-assembling fatty molecules that could form primitive cell-like membranes. There is the thiester world and the zinc world, each proposing its own specific chemical engine for getting the whole process started. Each of these ideas is genuinely useful. Each one is supported by real laboratory evidence, showing that the relevant chemistry is at least plausible under early Earth conditions. But here's the problem that has quietly haunted origin of life research for decades. None of them, on their own, manages to explain the entire journey. Each theory tends to solve one narrow piece of the puzzle while simply assuming the rest of the necessary chemistry showed up on cue. Scientists have effectively been left holding several different puzzle pieces from what appear to be several different puzzles, without a clear picture of how they might actually fit together into one continuous process. It helps to remember just how old this scientific pursuit actually is. In 1953, chemist Stanley Miller and his advisor, Harold Urey, ran what would become one of the most famous experiments in the history of science, sealing water, methane, ammonia, and hydrogen inside a sterile glass apparatus, then sparking the mixture with electricity to simulate ancient lightning striking a young, lifeless Earth. Within days, the mixture had produced amino acids, the basic building blocks of proteins, seemingly appearing out of nothing but simple gases and electrical energy. For a moment, it looked as though science had cracked the code of life's origin in a single afternoon. It had not, of course. Amino acids are a long way from a living cell, and over the following decades, scientists gradually realized just how enormous the remaining gap actually was. Producing the raw chemical ingredients for life turned out to be the easy part. Explaining how those ingredients organize themselves into something capable of storing information, capturing energy, and reproducing itself without any external guidance proved to be a vastly harder problem, one that has occupied entire scientific careers ever since without producing a fully satisfying resolution. Part of the difficulty is that the origin of life likely was not a single event at all, but a long, gradual accumulation of small chemical successes, stretched across many millions of years in countless different environments across the planet simultaneously. Reconstructing that kind of diffuse, slow-motion process from 4 billion years of geological remove is a bit like trying to reconstruct the exact recipe of a meal from nothing but a few crumbs left on an empty table. Researchers have real ingredients to work with. What they have lacked until now is a convincing account of how all of those ingredients came together in the right order, at the right time, under the right conditions. This is where Professor Yongdong Jin of Shenzhen University's School of Biomedical Engineering enters the story. Rather than proposing yet another standalone chemical starting point, Jin's newly published hypothesis asks a different kind of question entirely. What if the missing ingredient connecting all of these fragmented theories was never a specific molecule at all, but a specific kind of structure? one small enough and versatile enough to have done several of the necessary jobs simultaneously? Jin's answer centers on what he calls mineral nanozymes, or M-enzymes for short. He say are naturally occurring mineral nanoparticles, tiny clusters of atoms, just billionths of a meter across, made from materials like metal oxides, metal sulfides, and even trace noble metals. The word nanozyme itself comes from combining nanoparticle with enzyme, and it is a deliberate choice of language because these particles behave in a strikingly similar way to the protein enzymes that drive chemical reactions inside every living cell today, except these particular structures require no biology whatsoever to exist. They can form entirely through ordinary geological and chemical processes. This is not some exotic laboratory invention imagined purely for the sake of a new hypothesis. Scientists already know that nanozymes exist naturally, circulating in staggering quantities through Earth's oceans, atmosphere, and soil, playing a genuine role in modern environmental chemistry. What Jin is proposing is that structures very much like these were not simply a modern curiosity, but were actually present, active, and quietly doing chemical work on the primordial Earth billions of years before the first true cell ever assembled. The hypothesis paints an unusually vivid picture of what early Earth might have actually been doing during those long stretches of geological time. Rather than picturing a single dramatic moment where life sparked into existence, Jin describes the entire planet functioning as an enormous, patient, all-in-one natural laboratory, running the same reactions over and over across millions of different micro-environments simultaneously for hundreds of millions of years. Active volcanoes, deep-sea hydrothermal vents, and geothermal hot springs are singled out as especially promising environments. These locations naturally combine intense heat, sharp temperature and pressure gradients, mineral-rich fluids, and constant chemical turnover. Exactly the kind of conditions capable of forging fresh mineral nanoparticles directly out of raw, inorganic material. In fact, modern laboratory researchers already use remarkably similar high-temperature, high-pressure conditions to manufacture artificial nanozymes for industrial and medical use today, which gives the hypothesis a genuinely testable, real-world parallel rather than pure speculation. Once formed, according to the hypothesis, these primitive mineral nanozymes were not passive bystanders drifting through the primordial soup. They were active participants doing real chemical work. Jin's paper describes several specific jobs these particles may have carried out simultaneously, acting as catalysts that sped up otherwise painfully slow chemical reactions, physically binding and concentrating loose molecules onto their surfaces, so useful reactions could actually occur, shielding fragile early molecules from the destructive ultraviolet radiation that would have bathed the young Earth's surface long before any protective ozone layer existed, selectively favoring certain molecular structures over others and managing the flow of raw environmental energy, drawn from sunlight, heat, and even electrical activity like lightning, converting that energy into the kind of chemical information that could eventually be copied and passed along. That last point is arguably the most important one in the entire hypothesis. Turning raw environmental energy into something resembling storable, transferable information is widely considered one of the true hallmarks separating simple chemistry from anything resembling actual life. If naturally occurring mineral particles were already capable of performing that conversion long before anything resembling a gene or a cell existed, it would help explain one of the most stubborn gaps in origin of life science. How did purely random chemistry ever begin behaving in the organized, information-preserving way that biology absolutely requires? If a single tiny mineral particle quietly doing 5 different jobs at once sounds almost too efficient to be a coincidence, you are not alone in that reaction. And there is an even stranger twist to this story still ahead. Sticking with the video is the best way to see exactly where it goes next. Buried within Jin's broader framework is one particularly striking proposal that has drawn extra attention from other researchers in the field: the idea of an also world, named after gold's chemical symbol. Jin argues that gold nanoparticles specifically may have played an outsized role during this early chemical evolution, potentially becoming some of the most effective and important mineral nanozymes operating on the ancient Earth. This is a genuinely surprising claim because today, gold nanoparticles are almost exclusively associated with modern human technology, manufactured deliberately in laboratories for use in medicine, electronics, and chemical research. On their own, in raw form, free gold nanoparticles tend to be chemically unstable, prone to clumping together unless they are coated and protected by specific organic molecules. But Jin's hypothesis proposes a clever workaround rooted entirely in early Earth chemistry. Once other simpler mineral nanozymes had already begun producing basic organic molecules such as thiols and amines, those newly formed molecules could have coated and stabilized any raw gold nanoparticles they happened to encounter, effectively protecting them and allowing them to persist and keep participating in ongoing chemical reactions. In this reading, gold nanoparticles were not necessarily the very first actors on the scene, but they may have become an unusually capable and durable one soon afterward, once the surrounding chemical environment had matured just enough to support them. Beyond describing what these mineral nanozymes may have actually done, Jin's paper also lays out 4 broader conditions. The hypothesis argues were necessary for early life-related molecules to survive, stabilize, and eventually evolve into something more complex. The first involves wet-dry cycling paired with amphiphilic chemistry, referring to environments like tidal pools or shoreline sediments that regularly alternate between soaking wet and completely dry, conditions known to help concentrate loose organic molecules and encourage them to line K together into longer, more complex chains. The second involves self-assembly and self-organization, the tendency of certain molecules to spontaneously arrange themselves into ordered structures without any outside guidance, purely as a natural consequence of their own chemistry. The third involves catalytic and protoenzyme activity, essentially describing the specific chemical jobs the mineral nanozymes themselves were proposed to be carrying out. And the fourth involves pairing, symbiosis, and stabilization, the idea that different early molecules likely needed to cooperate and mutually reinforce one another's structural stability in order to survive and persist across time, rather than any single molecule managing the entire feat entirely on its own. Taken together, these 4 conditions function as something like connective tissue holding the broader hypothesis together, an attempt to explain not just where the necessary raw chemical ingredients for life might have come from, but why those ingredients would have actually stuck around long enough and cooperated closely enough to eventually assemble into something resembling a genuinely living system. What genuinely distinguishes Jin's proposal from the long list of origin of life theories that came before it is not any single new discovery, but its ambition. Rather than competing directly against the RNA world, the metabolism first model, or the lipid world for the position of correct answer, the nanozymes hypothesis tries instead to explain how several of these seemingly separate processes could realistically have been operating together at the very same time, driven by the same underlying category of tiny mineral catalyst. Under this reading, the RNA world and the metabolism first world are not necessarily rival explanations locked in permanent competition with one another. They may simply describe different downstream stages of the very same broader process, one in which naturally forming mineral nanozymes first got the whole engine of prebiotic chemistry running, before eventually handing certain specialized jobs off to more complex organic molecules as those slowly emerged and gradually took over. That kind of unifying ambition is genuinely rare in origin of life research, a field that has historically been defined by sharply competing camps, each one deeply invested in its own particular starting point. It is also precisely why researchers across the field have started paying close attention to this specific proposal, even while many remain understandably cautious about fully embracing it just yet. Whenever a hypothesis this ambitious enters a field as contested as origin of life research, the natural next question is how other scientists in that field are actually reacting to it. Unlike a single narrow laboratory result, which can often be quickly replicated or refuted, a broad unifying framework like this one tends to draw a more layered response. Part genuine interest, part professional caution, and that appears to be exactly what is happening here. The interest is easy to understand. Origin of life research has spent decades producing narrow, competing theories that rarely spoke to one another in any satisfying way, and a framework that offers a plausible mechanism for connecting several of them at once is inherently attractive to researchers who have grown tired of that fragmentation. The nanozymes hypothesis also arrives with a genuine advantage that many previous origin of life proposals lacked: an existing, thriving field of modern nanozyme research to draw on, built over the past 2 decades primarily for medical and industrial applications, which gives Jin's proposal a foundation of real, peer-reviewed chemistry to stand on rather than pure theoretical speculation. At the same time, seasoned researchers in this field have learned, often through hard experience, to treat any single unifying theory with healthy skepticism until it survives sustained testing. Origin of life science has seen confident, elegant frameworks rise to prominence before, only to be gradually narrowed, revised, or quietly set aside as new evidence accumulated. The RNA world hypothesis itself, once treated by many researchers as the leading explanation, has faced its own significant challenges over the years, particularly around explaining exactly how self-replicating RNA molecules could have formed spontaneously under realistic early Earth conditions in the first place. That history offers a useful reminder that even the most promising frameworks in this field tend to evolve substantially, or occasionally collapse entirely, once they meet the full weight of experimental scrutiny. It is worth being genuinely honest about exactly what this discovery does and does not represent, because getting that distinction right matters enormously in a field this famously difficult to settle with certainty. The nanozymes hypothesis is a theoretical framework, carefully built from existing chemistry, geology, and nanoscience research, and published through the ordinary peer review process. It is not the result of a single dramatic laboratory experiment that definitively recreated life's origin from scratch, and no researcher involved is claiming otherwise. Origin of life science is notoriously difficult to test with total certainty, for the simple reason that the actual events in question took place roughly 4 billion years ago, leaving behind no direct physical record for scientists to examine. Researchers can recreate plausible early Earth conditions inside a laboratory and observe what chemistry naturally emerges, but they can never fully prove that the exact same specific pathway actually happened on the real ancient Earth. This is precisely why the field has produced so many partially convincing theories over the decades, rather than one single universally agreed upon answer. It is also exactly why any new hypothesis, however elegant and unifying it appears on paper, still needs to be rigorously tested against future experimental evidence before it earns broader scientific acceptance. What makes the nanozymes hypothesis particularly promising, though, is that it is genuinely testable. Because artificial nanozymes are already manufactured and studied extensively in modern laboratories, researchers now have a realistic path toward directly testing many of this hypothesis's central claims, recreating plausible early Earth conditions and observing whether natura, life-forming mineral particles, genuinely can perform the catalytic, protective, and energy-converting roles the theory assigns to them. That reachable, testable quality is exactly what separates a genuinely promising scientific hypothesis from pure speculation. And it is a major part of why this particular proposal is generating so much serious discussion. Step back from the specific chemistry for a moment, the mineral nanoparticles, the gold nanoparticles, the wet-dry cycling, and something larger comes into focus. The search for life's origin is not simply an academic puzzle confined to dusty journals and specialist conferences. It is, in a very real sense, an attempt to understand our own most fundamental origin story, the actual starting point of everything that would eventually become every living thing that has ever existed on this planet, humans very much included. Every theory in this long, crowded field, from the RNA world to the iron-sulfur world to this newly proposed nanozymes framework, is ultimately reaching for the same basic answer. At some specific point, in some specific place, lifeless chemistry crossed a line it could never fully cross back over, and it began, however faintly and however slowly at first, to behave like something that was unmistakably alive. Jin's hypothesis does not claim to have found the final, definitive answer to that ancient question, and it would be dishonest of any science communicator to suggest otherwise. What it does offer is something arguably just as valuable at this particular stage. A genuinely new lens, grounded in real, testable chemistry, capable of drawing together decades of fragmented, competing ideas into a single coherent narrative that finally makes intuitive sense as one continuous process, rather than several disconnected explanations awkwardly stitched together after the fact. Somewhere in the deep, scalding waters near an ancient hydrothermal vent, or perhaps within the mineral-rich mud of a long-vanished tidal pool, unimaginably small particles may have quietly gone about the essential, patient work of turning raw rock and water into the very first flickers of something resembling life. If Jin and his colleagues are right, then in some genuinely profound sense, every living thing on this planet, every plant, every animal, every single one of us, ultimately owes its existence to structures smaller than a virus, doing chemistry nobody was around to witness, on a planet that had not yet become alive.